My interest in this topic, at the Land Institute goes way back. I was at a prairie festival in 2000, and it was before Lee was there as a staff member. I forget who was giving the talk on the stage. I brought students from Prescott College, and whoever it was described their version of how they saw the nitrogen economy of a perennial polyculture working out. And it basically amounted to having a grain legume that fulfilled maybe 25% of the cropping system, 25 to 50%. And that the nitrogen fixed in that grain legume, they worked out the math of approximately how much nitrogen that would pretty much satisfy the other crops' nitrogen needs. It was kind of just a back of the envelope suggestion of how at least the mass balance might work out. And I was very focused on this topic at the time and continue to be. And I was motivated to write a 2-page memo and stick it in every researcher's box at the Land Institute, completely unsolicited. And I thought, well, this is going to tell me something about the group and what they think of me. And so rather than just ignore me and never invite me back, I got an invitation 2 weeks later to participate in the Graduate Fellows Program at Matfield Green to dive into this topic. And basically the two things I was trying to point out is that the pulse, if you produce a legume seed in an intercrop and then you export that pulse, the vast majority N is contained in the nitrogen-rich compounds of that seed. And so it's pretty hard to pull off a high-yielding grain intercrop and supply the nitrogen needs of the associated grain, which is pretty hungry for nitrogen too. The other issue that I was pointing out is just how in traditional annual rotations, you have a kind of alternation between a high and demanding cereal that's followed the incorporation of a nitrogen fixing legume. And typically people will incorporate that nitrogen fixing legume, induce decomposition or mineralization, release of nitrogen, and then it raises the N status of the soil and the cereal takes it up, takes advantage of that, draws down some of the available N, and then that sets the scene for low N status when you once again plant the legume. That low nitrogen helps it fix its maximum amount of nitrogen, raises the N, incorporate it. So this pulsing happens in an annual rotation, but clearly that's not possible in a perennial polyculture. They're sitting there together, and you kind of you want both high and low soil nitrogen conditions to almost exist at the same time. That was the kind of paradox that I was floating back there in 2000. So now we advance 22 years, here we are, and this is one state of affairs, this Kernza alfalfa intercrop that you've heard some about. I went after alfalfa when I got to the Land Institute, partly because of potential, apparently likely resource partitioning with a deep alfalfa root and a heavy fixing forage legume in alfalfa, one of the most high fixing forages or legumes, period. And I went with some spacings that were a little bit wider than almost everyone has used in intercrop experiments that we've heard about. And I think you're going to see that actually has a pretty important impact and is kind of exciting. So just to review the design a little bit here, the intermediate wheatgrass alfalfa intercrop had row spacings at 38 centimeters between the alfalfa and the Kernza. Then I had a wide space wheatgrass and a narrow spaced wheatgrass, and by designing it this way, we're able to have an addition design where essentially the intercrop is a situation where inserting an alfalfa row, adding it to the wide space Kernza there. And then alternatively, the more narrowly spaced monocrop serves as a benchmark for a replacement design. So we're taking out a row of wheatgrass and replacing it with alfalfa. And there are two different ways of looking at an intercrop, but we learn things looking at them through these two different lenses. And I'll say that we alfalfa by hand regularly when it's 10%, as one does with alfalfa. And yet we have built this machine to do it on a larger scale at the field scale, mow it. And to me, this is one of the Achilles heels of what I'm talking about. Having to mow the alfalfa is a lot of fossil fuels to move that tractor through once, twice, 3 times a year. But you will not get nearly the end fixation in alfalfa or any unless you are harvesting it at the rate that it can reproduce. So that's a pretty important feature also of if you're trying to balance the nitrogen economy of the system to maximize nitrogen fixation. Brandon has gotten funding, and we're very slowly developing a forage harvester for an intercrop system as well, that you can actually remove the alfalfa rather than just lay it down as a mulch. So there's a couple of busy slides. Don't worry too much, because there's a much easier way to look at data that will follow these. But I wanted to put up just the overall results so you can see a pattern. These are years on the x-axis. This is total biomass, above ground net primary productivity on the y-axis. And we see a general trend down from year 1 to year 5, which is something we've talked about and seen in terms of some yield decline in wheatgrass. This is where the intercrop is. The dashed lines above the intercrop in this slide are the fertilized versions, with the wide space being the gray one and the narrow space wheatgrass being the blue dashed line. And then below the intercrop is the unfertilized treatments, right? So the kind of right in the middle of fertilized and unfertilized. Interestingly, and that's just wheatgrass biomass, not the alfalfa also. When you add the alfalfa with the intercrop design, you actually achieve levels of above ground productivity that are comparable to the fully fertilized 150 kilograms of N per hectare per year monocrop narrow space Kernza. No fertilizer in this treatment here in the intercrop. In terms of grain yield, the grain yields are low. This is cycle 2 Kernza, folks. And yesterday, Olivier said, you know, it seems like the yields persist longer if there's some stress in the system and they're not maximized yields. And that is certainly something we're seeing here. And I realized that the percent of wheat yield in this Kernza is almost the same as the percent wheat yield that Olivier was talking about in France. It's just their wheat yields are a lot higher. And so his Kernza yields, but they proportionally are not terribly different what we're seeing in these plots. What we are seeing is a level of stability across the 5 years that is pretty unusual, with no statistical decline compared to all of the other treatments in the intercrop system of grain. So the easier way to look at, or a way that I find really useful to look at these data are through a part of the land equivalent ratio. This is a ratio that people interested in intercrops use all the time to understand if the intercrop yields more than the monocrop. But to actually fulfill the whole ratio, you need monocrop stands of both of the species. And we didn't have monocrop plots of alfalfa in this experiment. And so we just have what are called relative yields of the wheatgrass in the intercrop versus the wheatgrass in the single species stand. And then I multiplied it times 100 just so because a lot of audiences can understand, is it 100% of the monocrop? Is it 150%? Is it 50%? So that's the kind of, when we look at these next 2 slides, that you should have. So these are the most interesting, if you want to know what the most interesting slides are of the presentation, the next two are they, okay. Oh yeah, well no, my part of the presentation, by the way. Let's be clear. So, and the above ground net primary productivity slide on the left, this is the addition design, okay? And so what we have is the percent of the monocrop yield of the wide space Kernza rows. These are 76 centimeter Kernza rows. What the red line, I can point at it, can I? Yeah. What this line shows is what happens to the yield over 5 years when you put alfalfa in between those rows. How does it compare to when you don't? And what we see quite clearly is, with the exception of the establishment year, there is what we would call facilitation. We have more wheatgrass production in biomass than we do without the alfalfa, okay? They're probably competing a little, but the benefits of the alfalfa exceed whatever competition might be going on between those species. In terms of comparing it though to the fertilized, actually I want to talk about this first, grain. The grain has a similar pattern, but not quite as dramatic. It's a little bit better until year 5 when it's a lot better. So year 5, clearly the unfertilized system is kind of hitting the wall and crashing. And these show to the greatest extent where we have nitrogen limitation and nitrogen sufficiency. Well, okay, we have these down below too. I actually drew these circles early this morning, so I forgot where them. The fertilized wide-spaced intermediate wheatgrass yielded higher than the intercrop in both, so I need to reset here. When you add alfalfa to the fertilized wheatgrass, you have lower biomass yields across the 5 years, except for year 1, interestingly, but not very low, okay? And so the intercrop competes pretty well with a fertilized monocrop. And when we look at the grain yield, though, it's particularly interesting. We see kind of significant end limitation. In other words, you have quite a bit more grain produced in the fertilized monocrop at wide spacing than the intercrop in the first 3 years, and especially those years 2 and 3 are pretty significant. Whereas in years 4 and 5, it catches up and the intercrop is actually yielding comparable or greater than the monocrop fertilized. So I think that this is the best illustration of the performance of the intercrop, the addition. When you start doing replacement, it gets a little tricky to interpret, but we can learn some things from it as well. So when you replace one row or every other row of monocropped Kernza with alfalfa, you will see an increase in productivity right off the bat. I mean, the establishment year is always an exception. We can talk about that. But thereafter, because of what found and limitation developing in the narrow-spaced Kernza, and this is, these are 38 centimeter rows, pretty wide. I mean, this narrow space is wider than many people's intercrop studies that we've seen. You still see much greater productivity in the intercrop than the monocrop. Similar with grain yield, except for the first year, you have much greater yields in the intercrop system than the narrow-spaced monocrop. When you add fertilizer, you have, not surprisingly, and by the way, the density of the intercrop, intermediate wheatgrass, is half in these slides what it is in the monocrop, right? And so it's not surprising, if they were competing equally, you would have 50% yield of wheatgrass in the intercrop compared to the monocrop, 50% as many plants in the intercrop as the monocrop. But that's not what we see. We see in between 50 and 100% of productivity of biomass in the intercrop compared to the monocrop in the fertilized replacement situation. But with seed yield, with grain, we're actually seeing greater yields starting in year 3 compared to the fertilized monocrop. So, I mean, that's exciting. It's very interesting. Let's see, is there anything else I need to say about the most interesting slide? These also are kind of interesting. In terms of stepping back and looking at the overall nitrogen economy of these systems, what does the mass balance look like kind of from year to year and on a cumulative basis? This is across the 5 years, where you take the managed end inputs and subtract the harvested exports. That's all we're looking at in these slides. How does it line up? And in year 1, we had actually a negative balance. All systems exported more N than were brought in, either through nitrogen fixing legumes in the solid blue line or fertilized systems. These are always going to be negative because there's no fertilizer inputs being applied or legumes. But we can see here the dynamics of having a legume intercrop. The nitrogen balance varies quite a bit from year to year, depending on the relative productivity of alfalfa compared to the wheatgrass in those systems. When you convert these to cumulative end balance, what we see is quite a bit of alignment in the first 3 years with the N fixation from the alfalfa and the high fertilizer input. And then though, because mainly of decline in yields in the monocrop high fertilizer system, you have a positive end balance. We're putting quite a bit more nitrogen at 100 kilograms per hectare per year into that system than is being removed. Now, it's quite possible that a significant amount of that nitrogen is vulnerable to loss, as we've talked about, because we're over fertilizing, right? But what we have to remember is that when we shift from a tilled system to a perennial system, we would expect carbon sequestration. We're going to start to, right off the bat, accumulate soil organic matter. And based on certain assumptions about what that might look like based on other conversion studies, here we estimated that about somewhere between 20 and 100 kilograms per hectare per year is being locked up in the soil, in the aggregating soil organic matter pool. And that comes to, again, over the 5 years, somewhere between 100 and 500 kilograms of N might be sequestered at a ratio of 10 to 15 to 1 carbons per nitrogen. There is a nitrogen cost to sequestering carbon, and it's got to be paid by that legume if it's in an intercrop system. And it's got to be paid by the fertilizer if it's in the fertilized system. I wondered if some yield decline might be competition with microbes immobilizing nitrogen later in time in a cropping system. Another interesting way of looking at these findings is just how fertile the intercrop is for the next crop. The non-fertilized systems are really drawn down. They have very low nitrogen availability at the end of this. But whatever crop is going into the legume, for example, or the fully fertilized system, it's quite a different context for productivity in terms of successional planning of the nitrogen economy. So just to go over the final digested outcomes of this study, the wheatgrass alfalfa intercrop, when compared to unfertilized monocrops in both addition and replacement designs, demonstrated consistent increases in grain and biomass productivity effect on productivity increased with time. The intercrop yielded comparable total above ground productivity as the narrow-spaced fertilized wheatgrass. It yielded less biomass than fertilized monocrop wheatgrass in 4 to 5 years, but matched or exceeded grain yields in the second half of the experiment in both addition and replacement designs. Maintained the most stable grain of any treatment across the 5 years. It had cumulative imbalance that suggests a high level of N accumulation with moderate synchrony between nitrogen supply and export and end storage. Apparent N limitation is evident relative to the fertilized treatments, especially in year 2 and grain yields. And we may be able to overcome N limitation in years 1 through 3 by altering management. And that some 3 ways that we might do that are starter applications of synthetic N. A recent study out of Sweden at how fertilizer might affect legume fixation when growing with Kernza suggests that the Kernza really takes up the soil nitrogen enough that you could get away with fertilizing a fair amount and still have N fixation taking place in an intercrop if that's what one wanted to do. Another idea would be to intercrop wheatgrass with alfalfa and a perennial legume such as red and white clovers that are more, they decompose and mineralize more quickly. They don't fix as much nitrogen, but they let it go a lot faster. And so that might release nitrogen earlier than alfalfa accumulates in the soil. And finally, the rotational intercrop is an idea that I've presented before. We're working on it at the Land Institute right now. We have some pretty neat trials in which we avoid this N deficit I've been describing in the first 2 years. That is inevitable when you plant a seed of alfalfa and a seed of Kernza. You just can't expect that alfalfa to deliver the nitrogen, deliver the goods in a very tight time frame. It takes years to develop that soil organic nitrogen pool. So why not front load that nitrogen first and then either make strips with an undercutter or herbicides, as we've done in this case, and sow directly Kernza into those undercut strips, leaving alfalfa in between. So you both have an effect of the rotation, like I said very early on, the mineralization that comes with incorporating a legume, but you also have strips of alfalfa that continue to pump nitrogen into the system after that. And the results that we've had look very promising, and I'm out of time. So I will be happy to talk with you about this experiment later, and maybe we'll swing by and take a look at it later on as well. So thank you much for your attention. And now it's Jess's turn. Ooh.
Oh, you have a tractor.
This has never worked on any other computer. This is applying the herbicide strips. And this is the, how did we get that to go? Here we go. Oh, okay, well, that was too good to be true.
Yeah, I think so.
Oh, no, there it is. So that's the undercut for people that wanted to see what that looked like. Okay. So now that I'm giving it to Jess, right? I'm just jumping ahead these slides. Come on. No, come on.
Oh, your video crashed it.
Oh, this is. Okay, so do I really have to restart this program? This is going to require upper division technical assistant. I can't believe that just happened.
Commercial break.
Oh, it's really unhappy, huh? Oh,
Okay, so yeah, that's it.
this one.
Yep.
Yeah.
Are you here? Where is?
Yeah.
Since you just
It's the next. Here,
What are you seeing over there? Aubrey, it's
Okay.
good?
Oh. Here, Tim's, I'm just showing your acknowledgements.
Oh, yeah,
That's
thank you.
And how do I, this is the little, let's see if we use this.
Do you need that laser thing?
I don't, I might keep it just in case. Keep it down there. All right, so Tim did a deep dive into legume intercropping as an example of how we might think about the ecological intensification of these systems. Like, how do we improve the nitrogen balance and use of nitrogen and ecological principles of nitrogen cycling and nitrogen fixation to improve these cropping systems. So now I'm going to kind of take us back out a bit of a level and talk about, how do we tell stories or communicate about the data we're collecting in terms of either of these frameworks, whether it's ecological intensification like Tim was just doing, or whether the ecosystem services that we're interested in. And so I'm going to use like 3 different examples of studies and types of data we're collecting to talk about this. And we'll see what you'll with. So the first example is something that we've probably talked about more than anything else with Kernza, nitrate in water. And this is a study that took place in Staples, Minnesota. It's has 27 inches of precipitation annually. It's on a sandy loam soil, and it traditionally has had, a lot of problems with nitrate in drinking water. So this is the location of this study, and situations like this have been the place of a lot of large Kernza plantings and a lot of this type of work. Oh, it says I'm connecting. Is it? Okay. What should I? Oh, okay. Screen 2. Okay. Are we back on? There we go. So treatments, corn soybean rotation, or a Kernza intercrop. And these had slightly different, they were both mineral fertilized, and they had different levels of mineral fertilization in both years of the study. And they were sampled with lysimeters. So this is a pretty common tool that we've been using to sample down into the soil profile. So these sandy soils really leach nitrate and leach nutrients a lot. They don't have a lot of organic matter. And so by using this lysimeter system, we can catch the nitrate that's leaching out of the system. So these are the results from 2 years. So I've kind of layered the different years on top of each other. So we have 2018. So this was Kernza and soy. The rotation the next year was Kernza and corn, and the year after that was Kernza and soy. And again, these were all fertilized with mineral water, and what we could see is, most of all, this beautiful result we always talk about with Kernza. So we have this change in nitrate and soil solution. So we have this gray line, and if this is pretty typical in a first year of Kernza stand, or in the spring where it's just getting started. And so the soil nitrate levels tend to be a little bit higher. But after this and through the remainder of this study, the nitrate in soil water is much lower in soil solution in Kernza versus the annual cropping systems. And so how would I tell this story? This is your turn. How would I tell this story if I want to talk about ecological intensification? So there's less nitrate in the water. What does that mean for the whole nitrogen balance that Tim was just talking about? Increasing our needs, right? And so this is another way of looking at all of that data Tim was showing, that we are keeping nitrogen, we are improving using the ecology of wanting to improve and tighten nitrogen cycling. We are improving our ability to grow crops on this land. Because if it's not in the groundwater, we hope that it is where the plants can get it. Yeah. So what would I say if I wanted to talk about ecosystem services? This is the easy one. It's not leaching into the water. What else? We're still getting, yeah, we're getting food out of the system. Yeah, and I'm so glad you mentioned that because that's the intersection of the two concepts, right? So if it's a better production system, it is provisioning food for us, or animal feed if it's dual use. Yep, absolutely. What else? Local wells, right? Water quality. So normally when I give this talk, my big headline is improved water quality. And I didn't do that today because I wanted you to say it, but yeah, so we talk about water quality. We are improving water quality for these municipalities, and that like framework and way of thinking like leads us into a whole other set of conversations we can have, because now we can talk about saving money for municipalities that don't have to mitigate nitrate. We can talk about incentivizing planting Kernza because it's going to provide all of these systems. The nitrate in water quality is what, you know, the big thing that people have been marketing for the first like seeds of a supply chain. Like this grain came from a place cleaning water in Minnesota. So this framework of ecosystem services can start to provide us some really powerful narratives to take this work to other stakeholders. All right, so the next example is aggregate stability. And the idea here is that, you know, as a soil naturally develops, you get these beautiful, I'm a soil scientist, so this is probably the most beautiful slide. To me, Tim, this is the best slide that I have. These beautiful aggregates starting to form and hold the soil together. Like this is a natural process of soil formation. And the example I'm going to show here is from a cropping system trial. So, you know, one is that people have been, you know, growing Kernza is in rotation because of this yield decline issue. And so we took advantage of that with this study and asked if Kernza compared to other cropping systems could be beneficial or could start to improve the transition time, like the 3-year transition time from conventional production to organic production. And so we did this the way that people typically would when you have higher corn and soy prices. So different systems for 2 years. We terminated all the Kernza or all the other systems, and we planted at the beginning of that 3rd year, we plant annual crops, because at the end of that 3rd year, 36 months on the dot, you can market those annual crops as organic corn and beans. So this is the study design. We replicated this at 3 different sites in Minnesota. So 3 different local climates, 3 different soil types. And we measured a whole heap of things. And this is, I'm not going to get into all this data, but this is just to kind of orient you to this study and let you know like some of the other data that's going to be coming out of this in the next year or so. So we measured a whole lot of environmental variables, including a whole list of different soil health indicators. And we also measured a lot of economical variables, including the grain yield, the biomass yield, you know, root biomass. I guess you could call that either environmental or an economic, weed pressure. And so we looked really holistically at these systems. Like, are they improving anything regarding the ecology of the ecosystem? And are they, in terms of the soil related factors? And is this strategy for the transition time also going to be beneficial to people using it? And so, you know, back to the aggregate stability, what we found is that the sizes of the aggregates started to change. And of all the things I measured, this is the most significant result that we saw. So 2 years to see these types of soil changes is a pretty darn time. And most of what we saw, the most significant things were in the sizes of these aggregates. The top panel is the mean weight diameter. And so we do this, filter all of the soil through a set of different sieve sizes to look at the sizes of both the amount of water stable aggregates and the sizes of them. And so the panel or the mean weight diameter of the aggregates, the real size across all the different size classes, and is kind of a narrowing in on just what % of our sample were the biggest aggregates, which means that they're kind of the most bang for your buck in terms of soil formation. And what we saw is that three, and then each of these set are the different cropping systems at our 3 sites. And what we saw were that Kernza and the Kernza alpha intercrop had the biggest mean weight diameter in St. Paul. And at our other site, we saw that it was mostly just the aggregates. And now we are actually seeing alfalfa, one of our other perennial systems, is also in certain situations improving the size of these aggregates. And then our other site, it was really back to just Kernza influencing this aggregate size class. And so now we're talking about aggregate sizes and soils. So if I want to think about ecological intensification, how to talk about this one? Yeah, so if you have more aggregate, that we have more organic that'll hold water. Yeah. Yeah, no, that's perfect. Right. So if we have bigger aggregates, right, it could mean that we have more organic matters, which means that we're holding nutrients, we're holding water, which will improve crop production, right, living life. And so the mineralization of the cycling of nutrients might be improved. What else? You're holding soil. Yeah, less erosion. So we're holding soil, right? So the physical, if we have more aggregation and water stable aggregates, it means that the likeliness that we're going to lose the soil in a big rain event is decreased. And so we're physically keeping the soil on the land and using the principle of a well-formed soil to improve our cropping systems. So what would I say if I want to talk about ecosystem services? Soil health, right? Our fun buzzwords of water quality and soil health. Yeah, so we're improving the soil health. So, and all the benefits that come with a healthy soil. Exactly. And with that, we are hopefully doing all the things that a healthy soil mediates, like water quality or like other benefits. All right, and so the last example is that I'm going to talk about our greenhouse gas emissions. And I'm going to prime us with kind of playing some Minnesota data off of some global data. So Minnesota data, this is a report from 2019. Actually, a new one just came out, but this is still pretty close to what we're seeing. And so every 2 years, Minnesota's been doing a greenhouse gas inventory. And as I was mentioning this new news article, this is one of the ways that Minnesota's been teeing up these big initiatives to mitigate climate change. And so you can see a couple of interesting things about agriculture, forestry, and land use here. So one is that it's really variable. So, you know, our cold weather in Minnesota really can influence the growing season year to year. But despite that, we still have about, you know, a third, maybe 25% of our emissions is in agriculture, forestry, and land use. And these are shown in equivalents. And so it's like every important, and I'll talk about the different types of important greenhouse gases in a minute, but this is kind of every type of greenhouse gas that's normalized to CO2, because CO2, carbon dioxide, is the most abundant greenhouse gas. And then you can also see, you know, energy generation, transportation, industry. A kind of goofy one in Minnesota, just for anyone who's curious, is our residential is quite a bit higher than the rest of the US. But otherwise, this is pretty, these are relative numbers of what you'd see across the US. And so we have this, you know, 25 to 35 million tons of CO2 equivalents per year is our greenhouse gas load from agricultural systems. And where does that come from? It comes from, and this is kind of zooming out. So this isn't just Minnesota anymore. These are global numbers from Project Drawdown. And so some of these things don't apply to Minnesota, but this is where our emissions come from. And so it's about 10% deforestation. I think that's probably not quite what we would see in Minnesota. A lot of that happens in tropical ecosystems. About 6% methane from animals and rice. So we don't have as much rice, but we have the animals for sure. about 4% nitrous oxide from fertilizer manure, and 5% other. And I put a note in here that this includes, when we talk about like straight carbon dioxide from tilling and from other sources of CO2 emissions. So this is kind of where our greenhouse gas emissions come from, and then how do these Kernza systems start to influence those greenhouse gas emissions. We're hopefully changing some of these things like the fertilizer use or the tillage. And so I'm going to show at first two different manure addition experiments. And so these are experiments, and we have one in Minnesota and one here at the Land Institute, where we're adding manure addition as the nitrogen source and as the nutrition source of the Kernza. And so these are just kind of the nuts and bolts of added, and this is the nitrogen in that manure, not the total amount of manure itself. And so in Minnesota, we had 80 pounds per nitrogen in 2020, 88 pounds per hectare in 2021. We had a similar treatment as ammonium nitrate instead of manure to make that comparison. And then we also, this is kind of a throw, wanted us to try this microbial inoculate, which I am usually highly skeptical of these, but I tried it. And I'm happy to chat with anyone later about like what they think about these different bacterial inocula for crop nutrition. But anyways, we tried it. And then in Kansas, there was also manure addition. And the different thing in Kansas was that they tried the manure addition in annual wheat in addition to Kernza. And so these are preliminary results, but what we are seeing is actually not a huge amount of change yet. And so even, let's see, so this is the Minnesota data. And with our nitrogen addition, we're actually not seeing, we're seeing a ton of variability, but we're actually not seeing a huge change in our nitrous oxide flux. Where we would expect to actually see a higher flux with the mineral fertilizer, but not seeing that necessarily. And so we could guess that in this situation, Kernza is still taking up all the nitrogen, not leaving it free to either be leached or to be blown off as nitrous oxide. As I didn't, the mechanism of nitrous oxide production is like highly correlated or related to some of the nitrate and leaching data that I showed before. Basically, if you have extra nitrogen, it's going to be leached, or some microbe is going to get it, either take it up, or if it's an anaerobic system, the microbes in the soil are the ones converting that extra nitrogen that we put on that system to nitrous oxide, which is this potent greenhouse gas. And so if we have systems where all that nitrogen is being used well, we would hope that we would both clean water and that we would remove nitrous oxide emissions. And so here, we're not really seeing a difference between these systems. That could mean that, Kernza, like this is very, very preliminary, but it could mean that in this situation, Kernza is not necessarily, it's still using that nitrogen that it needs. And in this study, we saw similar things in Kansas. And so even in the annual wheat, like we don't see, we see some emissions, but we don't see a huge difference between these systems. Maybe the no manure, the unfertilized treatment might be a there, but there's not a huge difference. And again, you could see a lot of variability and the fact that this is still a pretty new preliminary study. In other situations, though, we see different things. Oh, I forgot that I did show our CO2 fluxes also. So in this case, we actually might be seeing this increased CO2 flux with manure addition. And that for a couple of different reasons. It could just be from the extra respiration. So when we add manure and we're kind of juicing that microbial community, early in the years, we could be seeing that we just have more soil activity. CO2 and flux and soil respiration can be kind of tricky to interpret because because also can represent a healthy system, and you kind of need to start to look at the total carbon budget of these systems, interpret it well. But we are seeing some differences in CO2 flux that we need to kind of further digest. And to round us out here, oh, I am out of time. Well, Tim just showed you this, so I will zoom through the experimental design again and just show you the N2O emissions from the study that he showed earlier. And in this case, it's difference. And so where they had just intermediate wheatgrass, unfertilized intermediate wheatgrass with alfalfa, and these are the white bars are the first year and the dark bars are the second year. They didn't see a whole lot of difference in the N2O emissions. So again, the hypothesis would be that the Kernza is using that nitrogen really well. But they had just intermediate wheatgrass with nitrogen and phosphorus added. Now they see this, they were seeing this big jump in N2O emissions. And so, and this is what we would not necessarily hope to see, but in this case, like, could it be that we are either over fertilizing Kernza or not, you know, we don't have quite the tight nitrogen cycle that we would hope to have from an ecological point of view. So that is that's the greenhouse gas emissions. I know I stopped playing our game, but wanted to kind of dig into that theories of these emissions results a little bit more. So I just wanted to close with one parting thought to wrap up our whole talk. And that is that, like, kind of what we measure and the frameworks we use can really change the stories we're telling. And so again, I didn't do it in our last chapter of greenhouse gas emissions as much, but we can talk again about the conservation of nitrogen and the conservation of nutrients, or we could talk about climate mitigation, which is a really powerful narrative in a lot of states and has even gotten Kernza, you know, into this new initiative in the state of Minnesota. And so just that's how we are talking about these things can really influence the stories we tell. So thank you. And
Yes, we have time for questions. Yeah.
time for questions, but we will answer them. Yeah.
I just want the N2O study, the preliminary stuff, Jess, like, what was that experimental design again? I probably missed it. It makes a lot of sense to me, the results from Tim's new paper, that the addition of the nitrogen is contributing to more N2O. Talk about yours again. Like, what was that experimental design?
Yeah, so we had, oh, sorry. Yeah, we had Kernza planted, and it was Minnesota Clearwater. So everything you showed was cycle 2. So, and I'm not sure, I think over the last day or two, we've shown that the different cycles or varieties might be influencing some of these things, but it was just Kernza monoculture with these different additions. Manure or mineral fertilizer or nothing, or this bacterial addition. Yeah.
It was all some kind of external, like him seeing is that's where the losses are coming, but not with or lower than the intercrop.
Yeah.
Are you going to continue to monitor N2O there? And is this kind of like the totality of what we know or what has been monitored with Kernza and nitrous oxide?
Yeah, there has not been a lot. I think this is what we know. So
we are measuring it in that final rotational intercrop experiment also that I mentioned. So we'll have more data on that one in particular, but there's not a lot. There needs to be more.
Yeah.
Nitrous oxide is a big deal. And the idea of delivering the nitrogen in a slowly mineralized organic form, such as the alfalfa decomposing, as opposed to hitting huge slugs of available nitrogen and synthetic nitrogen is what we're seeing in you saw how much nitrogen is in that intercrop system. It's loaded, but it's producing nitrous oxide fluxes like the non-fertilized system. So that's exciting.
That is really exciting.
yeah, and we're we're measuring it in a couple of other we're continuing to measure it in a couple of other studies as well. We have a gas analyzer that kind of hits the three big gases, the nitrous, methane, and CO2. So hopefully we'll have more data along these lines to come.
Oh, Brandon, are we out of time?
We're kind of out.
Yep.
Hi, Jess, I have a question about the aggregate depth.
Mhm.
Were you using like 0 to 5, like scientific breaks, or were you using pedogenic breaks for like a forming new AP horizon?
Yeah, I guess we were going more in that frame of thought. I think we were doing around 20 to 30 centimeters, like the plow layer. So yeah, we were just looking in that changes in that most disturbed plow layer. So that would be more the kind of pedogenic classification of these soils than just the surface, which makes sense because the very surface is where you see most of the biological activity, but the soil is obviously forming a lot deeper, and Kernza's roots are forming a lot deeper. And so we try to go as deep as we can in the soil with just about everything we measure.
All right, thank you.
Thank you all.
Thank you.